Hi everyone, welcome to Thermo Fisher's 24 Hours of Stem Cells live training event. So my name's Dargitz and I'm a scientist here at Thermo Fisher Scientific. And a technique that I want to talk to you about today is picking pluripotent stem cell colonies. So there's a few different reasons you might want to do this. The first, and probably what most people think of, is just generally when your cultures start differentiating, you may need to go ahead and go in and pick to keep, which means selecting pluripotent stem cell colonies that have the correct morphology and will be capable of carrying on your stem cell line without differentiation. The second is in reprogramming. And so that's another common technique that researchers use and is really important as well. So you need to be able to go in and select the colonies that are going to become your clonal stem cell lines in that situation. And so really both of these two situations involve the exact same technique. And so that's what I'll go over today. There is a third situation where you might need to pick as well. However, this, instead of picking to keep, involves picking to remove. And that's actually physically going in and removing differentiated cells from your culture that may have spontaneously differentiated. And so what I'm going to do is go ahead and put these cells on the microscope, and we'll talk about what a good stem cell colony looks like and why you might want to select it when you're picking to keep. So these colonies all look good here, generally. You can see this kind of three-dimensional structure here is not something you'd want to pick. That's most likely differentiation. And so what I want to do is find a colony that's the right size and has the right morphology that we would want to go ahead and pick. So let's find what we can here. So this colony right here, you can see that there's some areas around it that maybe have cells that are somewhere in between pluripotent and differentiated. And so you want to make sure that you're only picking the cells that are fully pluripotent and will be able to carry on your line without any differentiation. And so I want to show this actual technique here by go ahead and using a needle that I've already prepared. So there's a lot of different tools you could use in this situation. Scalpels are fine, Pasteur pipettes, anything like that. In this case, I have a 25 gauge needle on a syringe here. And what I'm going to do is just go ahead and cut this colony into a grid pattern. So I'm going to come in and go one direction and then do the same thing after turning the dish 90 degrees. And then we'll go ahead and talk about what we're going to, how we're going to remove those pieces of the colony and add it to the next dish that we'll be using. So as I carefully uncap my needle here, I'm going to go in. If you are using a needle, it's important to remember to keep your bevel down. And then you got to make sure that keeping your colony in the center of the plate here. So you can see, pardon me, you can see the tip of my needle right there. So you really want to just be careful and make nice parallel cuts. I kind of by habit am cutting around the edges here. That's not so important when you're working on a feeder-free system, but if you're working with a feeder-dependent system, it's important to cut around the edges, since sometimes those feeder cells can adhere to your colony, and if you don't cut around the edges, you might have problems getting them off. So as I mentioned, I'm just going to turn this 90 degrees so that I can then make perpendicular cuts to the ones I've already made. And again, it doesn't have to be perfectly perpendicular. You can see these are at a slight angle, and that's all right. The key with choosing whatever tool that you're using is just making sure that you're not, it's not so thick that you're going to be having problems with killing a lot of cells where the actual needle is going over it. So what I'm going to do now is take my P200, and again, this is something. All these different steps, I encourage people to see what really works for them best. I prefer to use a P200 since the size of the bore is such that it's not so small that it's going to break the colony pieces into any smaller pieces, and it's not so big that I don't have very good motor control and I'm not able to get what I need. So I'm going to actually go in here with my my P200 and just kind of very gently lift those pieces off. And so you need to be careful that I'm only picking up the pieces from this colony, and then aspirating. So sometimes they might stick a little bit. You need to go in and loosen them off. But as you can see, for the most part, I've got everything off of the plate there. And then I'm just going to go ahead and move this to my next plate. Now, it's important to note that if this were in a reprogramming context, that colony would be starting its own clonal line. So only that colony would be going into a new well. If I'm picking to keep in a typical culture setting, then I would need to put multiple colonies from this well into a new well so that there's more material to grow from. So let's see if we can go ahead and find another colony that looked good, or maybe even some differentiation that we can remove, so I can show you that technique as well. Here's another good looking colony. So I'm going to go ahead and do the same technique. So as you remember, I'm going in here with my needle and just making one set of cross-hatched lines. So that was not as straight on that one. That'll happen sometimes, and that's totally okay. You just need to keep making your cuts and not worry about it. Excuse me. Oops, sorry. Hit the edge there. And sometimes on these it can be tough. You can lose where your actual tip of your needle is. Oops, sorry. Oops, I'm having some trouble here. I think I'm going to actually switch the tip of the needle. Sometimes those can get dulled a little bit. So I'm going to put that in the sharps there and grab a new one. And so, as I mentioned, picking to remove is important too. So after we do this, I'll see if I can find some differentiation. And then we can go ahead and scrape that off. And typically what I use for that is the P200. So again, lines don't look quite as straight as they did on the last one, but that's okay. We're still making this into a smaller pieces that we're going to be able to effectively pick up. So again, I'm kind of cutting over the edge here. Oops. There we go. So not my best work, but you can see there's still pieces here that we're able to pick up, and actually then we're going to just go ahead and turn that 90 degrees and do the same thing again. Okay. So that looks a little better. You know, sometimes they do get bunched up a little bit, and that's definitely possible that that may cause a little differentiation when it sits down, but again, that's why you want to make sure, whether you're doing reprogramming or just picking to keep normally, you're doing this several different times and picking lots of colonies, because if one doesn't work out the way you want it to, or it doesn't sit down for any reason, you should be able to do multiples so that you're successful. So now I'm just going to go in again with my P200 and kind of gently ease those pieces of the colony off the plate and aspirate those up as I do it. There may be some little pieces here stuck, and that's okay. Those are most likely too small to sit down and form a colony anyway. So now I'm just going to add that to my plate here. So I mentioned picking to remove, and again, I think that generally picking to keep, or picking your pluripotent colonies, is a much more common technique, but it's important to know both. And I would say even picking to remove is a little bit easier, since really all you're doing is just scraping those cells off the plate, and then you'll end up rinsing and removing them. So let's see if we can find a little differentiation here real quick. So again, I mentioned some of these other areas that are somewhere in between fully differentiated and pluripotent. Something like this where you can see the difference between cells. You can actually see space between the cells, whereas the normal pluripotent colony has that really tight morphology, that refractive edge, and a high nucleus to cytoplasm ratio. So something like these, again, a little more what we're looking at removing. Sometimes it'll look just like a fully differentiated somatic cell, and sometimes it'll just be like a colony that's started to look a little different. So we can go in here and just remove. And then, as you know, so now these cells are floating in the media itself, so you need to make sure that you go in and rinse the media several times before you're done with it, because if you leave those differentiated cells or if you passage them, they're just going to end up in your cultures going forward. Great. So with that, I just want to summarize things I talked about. So when you're picking a keep, there's basically two major reasons you'd want to do that. One is reprogramming, so you're selecting a pluripotent stem cell colony that's going to become your clonal cell line. The second is when you have a fairly differentiated dish and you want to select from it the pluripotent cells and pluripotent colonies that you want to go on and continue that cell line. And then lastly, if you have a fair amount of differentiation as well, you may want to go in there and actually physically remove the differentiation instead of selecting the pluripotent cells that you have. And so those are the three main areas. And again, that criteria that we look at for selecting the pluripotent stem cells when we do is that refractive edge of those colonies, the high nucleus cytoplasm ratio, and those colonies should be really tight with cells that are difficult to distinguish from one another. So with that, I'm just going to take any questions that there might be, and we'll move from there.
Thank you very much for that demonstration, Carl. I'm sure our viewers are a lot more confident about picking colonies after watching that. Of course, there are things that participants may want to dig into or clarify. And so, my name is Joanna, everyone. And like Carl, I'll work on the Thermo Fisher stem cell products. I will be moderating today's Q&A session. So please, if you haven't yet, please type in your questions into the chat and I will read them for you. Carl, the first question, which was sent in earlier, is this. What magnification are you using on your microscope?
That's a good question, Joanna. So this is actually a 5x. I've seen people use 10, but in general, for something that involves such fine motor skills, I find that a higher magnification makes it easier. Again, though, I mentioned this with several things, including the different tools you're using to do the actual picking and things like that, people should really try whatever they can and see what works best for them. If you like doing it at 20x, which is something I'm not comfortable with, and that works for you, then by all means you should do it. So I really encourage people to try a lot of different things and see what works, because this is, as I mentioned, a really important technique that you're going to be utilizing a lot.
Right. And here's another question from the same participant. Why can't you just aspirate the entire colony using a P200 tip? Is the segmentation necessary?
Sure. So if you were to go in and remove an entire colony with a P200 or aspirate it with a P200, you're going to get very heterogeneous sizes of the chunks from that colony, which can be a lot more problematic. Generally, as long as you're comfortable with the technique, you should be able to get similarly sized colony pieces, and that'll make for a more consistent culture going forward. So you could do that, but I think in general you're going to wind up with a much more heterogeneous population and more likely to differentiate and less likely to have your colony pieces sit down.
Right. And this is going back a little bit. You showed obviously how to pick the colonies, but how would a colony be identified by a novice, an appropriate one for picking?
Right. So as I mentioned, there's kind of like three main things you want to look at with pluripotent stem cell morphology in general. The first is how those cells are really tightly packed together. You can't really distinguish one from another. When they start to separate from each other, that's usually a sign that they're on their way towards differentiation. The second thing is that they have a tight refractive edge. And so that's something you can identify on the microscope itself. And if you want, we can go back to the screen at any time, but you can see colonies like this one here in the center of the microscope screen really have that distinct edge that's completely separate from the substrate itself. And you can easily identify where the colony starts and begins. This can be a little more difficult if you're using feeder cells, but that tight refractive edge should still be there. The last thing is high nucleus to cytoplasm ratio, which oftentimes you may have to zoom in on the colonies to really see, but that's something to keep in mind still. But I'd really focus on those first two things, the tight refractive edge and the tight colony where the cells are very close together.
And do you have any advice for reducing the spontaneous differentiation in the first place?
Absolutely. So that's something that's really important to do, because if you don't have differentiation, then obviously you don't have to remove it, which is great. The main thing that I would do in that case is be very consistent with your feeding, depending on the media you're using, but most people are feeding every day at the same time. You never want your cells to get the media to get acidified. That's a common cause of differentiation. You want to also be consistent about removing any small amounts of spontaneous differentiation that are occurring in your culture each passage. If you skip a passage, then it's more likely for that to build up over time and eventually take over your culture. So that's another big thing. The time out of the incubator is also very important. If you're leaving your cells on a microscope for a long time, their temperature might be dropping, and that is a common cause of differentiation. All these things really come into play, but in general, you just want to be very careful and take really good care of your stem cells, much more so than you would with any other type of cell, because they're very finicky.
All right. Here's another question. Do you ever use the aspirator to clean a plate?
That is something I have done in the past. I know some microscopes may have like a marker on the bottom or something like that, where you can actually physically mark places of differentiation. That's a great technique if you have access to a marker like that or something. Usually there's a circle or something of that nature, and so you would have to go and physically mark all over the plate. If you are doing that with a plate that's very, very differentiated, it's really going to take a long time, and so you might be better off picking to keep. But if there's a reasonable amount of differentiation that you can mark and then go in and actually aspirate, that's a great technique as well. We don't have that on this instrument, but that's another option that people can use.
And how do you remove differentiated cells just around the edge of a colony?
If there's cells just outside the edge of the colony, how do you remove them? So typically what I would do is then go into the center of the colony and only cut the middle out. If I'm worried about accidentally grabbing some cells from the outside, I would take just the very inside of it, because as you can see, sometimes it's a little difficult to get, the whole colony the way you want it and not get anything from the outside.
And say you did accidentally include some parts of the differentiated sections, how do you fix that mistake?
Sure. Again, that depends on whether or not you're doing a reprogramming picking or picking to keep in terms of just a normal cell line. So if you're keeping it for a normal cell line, you're picking, say, 5 or 10 colonies that are all going into the same plate, and you get a little bit of differentiation. That should be fine. In a reprogramming setting, that's going to have a larger effect. So as long as you have plenty of colonies to choose from, I would go ahead and then aspirate that well and not use that colony if you picked up differentiation.
This one is changing the angle a little bit. What are the benefits of hand picking over a bulk passaging reagent that may distinguish between different kinds of cells?
That's a good question. So typically, as long as your differentiation levels are relatively low, you really want to make sure you're doing bulk passaging, because again, that's going to be overall more consistent and allow you to do things much quicker. So your workflow will be sped up quite a bit. The problem occurs when you have a lot of differentiation and you're not able to pick to remove all of the differentiation in the plate. As long as you're able to remove everything in the plate in a relatively short amount of time, you're fine to do bulk passaging. However, if there's so much that you feel like the differentiation has sort of taken over, then that's when you'd go in and actually take the pluripotent stem cell colonies out and pick to keep.
Right, and that's just thinking about the maintenance of the pluripotent stem cells, right? There are specific applications where you need to pick specific colonies.
As I mentioned in reprogramming, you don't have that luxury unless you don't care about clonal cell lines at all. But most people, when they're doing a reprogramming, want to isolate individual clones that formed those colonies. And so that's when you would have to be very careful about only selecting one colony at a time that goes into one well at a time to form those clonal cell lines.
And so in the case where someone is required to pick their colonies, is it possible to pick colonies without the use of a cabinet screen microscope?
It is there. I have seen setups where you're in more of a dissection scope. However, mostly you want to stay within a laminar flow hood because the chance of contamination becomes very high if you take it outside of it. In general, even within a laminar flow hood, this is your most likely time the contamination will occur, and so it's a really time you need to be really careful and make sure. And that's something actually I didn't mention during the demonstration that I probably should have. So I want to hit on that point a little more. This is the most likely time that contamination will occur. You have your plate open, and so you need to be careful about passing your hand over the edge of the plate. And because typically stem cells are cultured without antibiotics, it's even more likely that will occur. So you have to be very, very careful throughout this entire process.
This is sort of related just a little bit. What is the maximum time that you'd be comfortable leaving cells outside the incubator?
That's a great question. And as with all these, it does depend on the specific cell line you're working with. But I wouldn't want to have them out for more than 20 or 30 minutes at a time. So I would say, you get them out, look at them, and then kind of start a timer or something like that, and be aware of how long it's taking. And if it ends up being that you need more time than that, I would recommend going through, if you have other plates, and doing that first before bringing those back out again. You don't want your temperature to be consistently lower than 37 degrees centigrade for too long.
Excellent. We are seeing a number of thank you messages here too, just so you know, Carl.
Great.
They're finding this very helpful.
Good.
Here's one question. When picking colonies, how many pieces should each colony be separated into, and how large should each piece be?
Sure. Also a very good question. So how many pieces they're separated into really depends on the size of the colony. So if you have a smaller colony, that'll be fewer pieces, and if you have a very large colony, that'll be many more. In general, if the colony is so small that you can't make more than into more than 4 pieces, I generally would say you shouldn't be picking it, because most likely that colony is not ready to be picked. As far as the size of the chunks themselves, I think that it's important to try to be as consistent as possible, colony to colony, and really it's more of a law of averages. So you need to find the kind of middle ground between so large that it's going to sit down and fold over on itself and cause differentiation, and so small that it actually won't be able to sit down and adhere to the plate. So something in the middle generally is what you're looking for. And so basically, if you saw what I was cutting, that's about the right size. But again, I think this is something that will just take practice and depends on what you're doing and what works for you.
And you showed not just picking of colonies, but the removal of, differentiation. How much differentiation necessitates either removal of those differentiated areas or picking a colony in order to keep it?
Sure. As long as it's noticeable differentiation, I would recommend removing it. As far as how much actually necessitates picking to keep, I'd say if you have more than 20 to 30% of your plate that's fully differentiated on a feeder-free system, that number's a little higher on a feeder-dependent system. But if you have more than, say 40% on feeder-dependent, that's when you probably need to go in and actually select your pluripotent cells to continue the line. That means the differentiation is starting to take over. Great.
And this is a question that goes into kind of reagents. I'm not sure if I'm understanding it correctly, so if not, please send in a clarification. But do you ever have to add inhibitors as you pick colonies? I imagine something maybe like a ROCK inhibitor?
Sure, that's something you can do if it's necessary. We typically don't do that just to kind of keep the cells in their normal state where they're not being perturbed at all. But that's something that if you're having trouble with viability after picking colonies, that you can give it a shot. So.
And more generally, a question on how many colonies typically grow per dish?
I don't know an exact number, but hundreds of colonies, depending on the size of the dish, of course. We typically culture ours in either 6-well plates or 60-millimeter dishes. In both of those, there would be hundreds of colonies. And also, it's worth keeping in mind that the colonies will start to grow together, especially in a feeder-free system, by the time they're about 70% confluent, which is when you want a passage. And that's something that later today in another one of the live trainings that Chad will be talking about in general with bulk passaging is the confluency and when to passage. But yeah, many hundreds of colonies will be growing in one plate at a time, and they'll start to grow together in a feeder-free system by the time they reach 70% confluency, which is when you would typically passage.
Another question just came in. What do you do if you accidentally cut a colony into uneven or very small pieces?
Sure, that's a great question. Generally speaking, I would say you want to just go ahead and press forward and use the material that you have, so long as you didn't pick up any differentiation in it. That said, if it's the cell colonies have been completely broken into very, very small pieces, they may not sit down. But unless that's the case, I would say you want to just go ahead and move forward and take the colonies anyway and see what happens. And that'll also inform your picking later to see what actually is capable of sitting down and forming pluripotent lines and what isn't.
And one final question, which I found very interesting. They're apologizing because this might be too far-fetched, but could we engineer new plates so as to reduce the technique error? Maybe pre-divided quadrant plates, or which could be picked with colony, or could help us pick the colonies.
Sure. I've definitely heard of micro-machine plates for different applications, usually in a differentiation context where one colony will grow and then they'll differentiate that into a specific cell type. But something like that could certainly work. I do think that it's important to make sure that the media is probably shared between them, as there's a lot of growth factors and other things that cells are spitting out, especially if you're working with feeders. On feeder-free, that may not be the case, but something like that could potentially work for sure.
All right. I think that's a wrap for today's live training event on picking colonies. Thank you again, Carl, for demonstrating, and thank you everyone for joining us here. 24 hours of stem cells does run for many more hours today, with scientific talks and many Thermo Fisher resources at your disposal. If we didn't get to your question, there are other opportunities to ask those in our booth, and perhaps those questions could be answered through our other training sessions as well. Thank you again.